Welcome class. In this session, we'll be discussing regarding the last step of aerobic respiration, that is electron transport chain, which is also called as ETC. So this electron transport chain, it is always coupled with the release of energy, and this is the step where exactly the oxygen molecule will be actually utilized. Okay, so electron transport chain can be defined as it is a metabolic pathway through which electrons from the high energy compounds such as NADH and FADH2 passes from one electron carrier to the another, which is coupled with the energy release. So this process is called as electron transport mechanism or electron transport chain. And this particular electron transport mechanism, it occurs in the inner mitochondrial membrane. So if we see in case of inner mitochondrial membrane, they will be having certain infoldings called as cristae. And this cristae, it will be having certain electron carriers, so which will be catalyzing this particular mechanism. And as we see in case of inner mitochondrial membrane, so mainly it is made up of 4 different electron carriers or 4 different complexes, such as NADH dehydrogenase, which is also called as complex 1, and this NADH dehydrogenase, so it is composed of a flavoprotein called as FMN, that is flavin mononucleotide. Similarly, it is composed of several molecules of ferrous sulfur protein or the iron sulfur protein. So these two are the components of NADH dehydrogenase. And the second compound is the succinate dehydrogenase, that is also called as complex 2. And this complex 2, it will be associated with the FAD molecule. So it is coupled with the FAD, reduction or oxidation of FAD. So that is complex 2. And complex 3 is called cytochrome B-C1 complex. So which is made up of the cytochromes. So it consists of cytochromes such as cytochrome B, cytochrome C1, as well as the iron sulfur proteins will be present, or the ferrous sulfur protein will be present in addition to this cytochromes. And this cytochromes, they will be having the ferrous ion or the ferric ion as a metal center. So next, coming to the fourth complex, that is cytochrome C oxidase. So this cytochrome C oxidase, it is made up of 2 cytochrome, that is cytochrome A1 and cytochrome A3. And these cytochromes, they will be having the copper as a metal center. It is also called as complex 4. So these are the fixed electron carriers that are associated with the inner mitochondrial membrane. So in addition to this, there are also 2 mobile electron carriers. So what are those? We'll study. We'll look into through this diagram. So this is the intermembrane of the mitochondria. So here you can see the complex 1. So that is NADH dehydrogenase. So this is complex 2. And then, so this is complex 3, that is cytochrome B-C1 complex, and this is complex 4, that is cytochrome C oxidase. So in between these cytochromes, if you see, so these are the fixed electron carriers which are fixed in their position in the inner mitochondrial membrane, and electron from one complex to another complex, it is carried out through the mobile electron carriers. So there are 2 such mobile electron carriers. So one is called as coenzyme Q, or it is called as ubiquinone, and another one is cytochrome C. So these 2 are the mobile electron carriers. And this ubiquinone or coenzyme Q, it is a mobile electron carrier which carries electron from complex 1 to complex 3, similarly from complex 2 to complex 3. Okay, so that is the electron carrier which is between complex 1 and 3, whereas cytochrome C is the electron carrier between complex 3 as well as the complex 4, which transport the electron from complex 3 to complex 4. So that is regarding the complexes or the electron carriers that are present in the electron transport chain, and all this will be present in the inner mitochondrial membrane. So next we'll discuss regarding the electron transport mechanism. So as it is said, so it is a metabolic pathway which is concerned with reduction of high energy electrons from the high energy compounds in order to release energy. And this electron transport mechanism, so it will be undergoing, so in this process we can see a series of redox reaction or the oxidation reduction reaction, where the electrons released from the high energy compound it undergoes series of oxidation as well as reduction, and finally that will be coupled with the energy release. So that is regarding the electron transport mechanism. So we'll look into the electron transport mechanism in detail. So first, the high energy compound that is produced is one is NADH and FADH2. So electron from these two will be reduced, okay, into a low energy electron, and that will be coupled with the ATP synthesis here. So first, NADH, it undergoes oxidation. So oxidation of NADH, it is coupled with release of the hydrogen ion as well as the electron, and the electron, high energy electron that is released upon oxidation of this NADH will be first accepted by the FMN, that is flavin mononucleotide present in the, so this is complex 1 components of, so this is complex 1. So it will be accepted by the FMN molecule, okay, and it gets reduced into FMNH2, okay, and the hydrogen ions that are produced, so it will be released into the intermembrane space. So these are reaction occurring in inner mitochondrial membrane, and in between outer and inner membrane, the intermembrane space will be present. So hydrogen ions that are released here upon oxidation of NADH, it will be pumped to the intermembrane space. So, and the electron will be accepted by the FMN and it gets reduced into FMNH2. So once the FMN is reduced, so now it gives its electron to the mobile electron carrier, that is coenzyme Q. And for coenzyme Q, in order to accept the electron, so it also withdraws 2 hydrogen ions from the mitochondrial matrix. So along with 2 hydrogen ions, it accepts the electron that is released upon oxidation of FMNH2 into FMN. The coenzyme Q will be reduced into QH2. So this is the reduced form of coenzyme Q. So now this reduced coenzyme, it will carry its electron to the complex 3. So this is the complex 3 actually. These are the components of complex 3. So it will transfer the electron to the complex 3. So in case of complex 3, so first cytochrome B. So I said both cytochrome B as well as cytochrome C1, they will be having iron as a metal center, okay, or the ferrous ion as a metal center. So electron released from the coenzyme Q, it will be accepted by the ferric ion, where it gets reduced into ferrous ion, and this electron from the ferrous ion will be further transported into cytochrome cytochrome C1 of the complex 3. Okay, so as electron is transported into cytochrome C1 of complex 3, now the ferrous ion will be oxidized into ferric ion, and here in the cytochrome C1, upon accepting the electron, the ferric ion that is present in cytochrome C1 will be reduced into the ferrous ion. So that is regarding the reaction that is occurring in case of cytochrome B-C1 complex or in the complex 3. And here, initially when the electron is accepted by the coenzyme Q, it also has taken 2 hydrogen ion from the matrix, and when they are transferring the electron to the complex 3, so this hydrogen ion will be pumped to the intermembrane space in the mitochondria. That is the space present between inner and outer membrane. Now, once the electron is transported to the complex 3, from the complex 3, now the electron will be transported into the complex 4 through the one more mobile electron carrier that is called as cytochrome C. So this cytochrome C, again, it has a ion as a metal center. So upon accepting the electron by the cytochrome C, so it gets reduced into the ferric ion present here will be reduced into ferrous ion, and this reduced cytochrome C, now it carries its electron to the complex 4. So in case of complex 4, actually copper ion will be there. So cupric ion will be present, which will be reduced into cuprous ion. Okay, in case of complex 4, where it consists of two cytochrome, that is cytochrome A1 as well as cytochrome A3. Both will be having the cupric ions as a metal center. So upon accepting electron, so here in the cytochrome A, the cupric ion will be reduced into cuprous ion, and then again it will be transferred to the cytochrome A3, where again here the cupric ion will be reduced into cuprous ion. And as here the electron is passing through this series of electron carrier. So during transfer of electron from cytochrome C to complex 4, again 2 molecules of hydrogen ions will be released into the mitochondrial intermembrane space from the mitochondrial matrix. Okay, and once this series of reactions are taking place here, as electron is transferring or as electron is moving from the complex 1 to complex 4 here, the energy of the electron, it keeps on reducing, and finally once the electron comes out of the complex 4, so now that electron will be a low energy electron or the weak electron. So this electron, it is called as weak electron, so which cannot be reduced further. And such low energy or the weak electron will be finally accepted by the oxygen molecule. So along with that, it also accepts 2 hydrogen ions and it will get reduced into a water molecule. So this is where 6 molecules of water will be formed during the process of aerobic respiration, and 6 molecules of oxygen will be utilized. And as here the low energy electron is accepted finally, so as this low energy electron is accepted by the oxygen finally, so this process is called as terminal oxidation, and here the oxygen is considered as dump yard of low energy electrons. So that is because further these electrons cannot be reduced. So hence the oxygen acts as a terminal electron acceptor, and the process is called terminal oxidation. And as all the low energy electrons are accepted by the oxygen to form a water molecule, oxygen is also considered as dump yard of low energy electron. So that is with respect to NADH molecule. Whereas if we consider with respect to FADH molecule, so this reaction will be coupled. So this reduction of the energy released from the FADH2, it starts from the complex 2. So complex 2 is called as succinate dehydrogenase, where succinic acid, when it is reduced into fumaric acid, so FAD will be reduced into FADH2, and the electron from this FADH2. So now this FADH2, it undergoes oxidation in the electron transport chain, and the electron released from this FADH2, it will be accepted by the coenzyme Q. Along with that, it also accepts 2 hydrogen ion, and further the electron from coenzyme Q will be transported to complex 3, and from complex 3, it will be carried by the cytochrome C to the complex 4, and from the complex 4, the low energy electron that is released, it will be accepted by the oxygen molecule, resulting in the formation of the water. So that is regarding the electron transport mechanism that is occurring in case of the during the aerobic respiration. And here nowhere we have discussed about the production of the ATP, right? So here the production of ATP, it can be explained to the process called as oxidative phosphorylation, and this mechanism of oxidative phosphorylation, It is explained through a process or the hypothesis called as chemiosmosis. So same process has been discussed in case of photosynthesis also. So here, according to chemiosmosis, so osmotic movement of the hydrogen ions across the membrane, it will be coupled with the ATP synthesis, right? So in case of here, it is called oxidative phosphorylation because ATP molecules are synthesized in the presence of oxygen molecule. So that's why it is oxidative phosphorylation. So if ATP is synthesized in the presence of light energy, then it is photophosphorylation, which can be seen in case of photosynthesis. So here in the respiration, we can see oxidative phosphorylation. So in case of the inner mitochondrial membrane, if we see the cristae of the inner mitochondrial membrane, it will be having a small lollipop-like structures called as F0, F1 particle, or these are also called as ATPase enzyme complex. So here, during the electron transport chain, we have seen that more number of hydrogen ions, it will be pumped to the intermembrane space so that the concentration of hydrogen ion in the intermembrane space, it will be more compared to that of the mitochondrial matrix. So as a result, what happens? So these hydrogen ions, they tend to come back to the mitochondrial matrix, but here the problem is your inner mitochondrial membrane, it will be impermeable for this hydrogen ion. It won't allow the hydrogen ion to pass through it. So in order to favor the movement of hydrogen ion, so there will be a complex 5 that is associated with the inner mitochondrial membrane, that is called as ATPase enzyme complex. So this ATPase enzyme complex, it allows the hydrogen ions to pass through it. Suppose if hydrogen ions has to be pumped from the intermembrane space to mitochondrial matrix, only option is they have to be transported through the ATPase enzyme complex or the CF0F1 complex only. So here, as the hydrogen ions are moving through the ATPase enzyme complex, so for the movement, so it generates the proton motive force. So that is energy which is generated due to diffusion of hydrogen ions from the intermembrane space to mitochondrial matrix, and the energy released by the proton motive force, it will be sufficient enough to add phosphate to the ADP to form ATP. And according to chemiosmotic hypothesis, it states that for every 2 hydrogen ions that is pumped from the intermembrane space to the matrix, so 1 ATP molecule will be synthesized. So that is regarding the oxidative phosphorylation. So hydrogen ions, as they are moving to the mitochondrial matrix, it generates proton motive force and it will be coupled with the ATP synthesis. So that is regarding oxidative phosphorylation. And here, this hydrogen ions are released when NADH as well as electrons from NADH and FADH2 is released, right? So hence we'll see how much ATP molecules are produced for one NADH molecule as well as for one FADH2 molecule. So we'll come back to this electron transport chain here. So if we look in case of the oxidation of one NADH molecule. So once the one molecule of NADH is oxidized, so in the 3 regions, 2 molecules of hydrogen ions will be pumped, right? So one is in the site 1, one in the region of site 3, complex 3, and third will be in the region of complex 4. So as a result, one NADH molecule, so it results in pumping of 6 hydrogen ions to the intermembrane space. And once the 6 hydrogen ions are coming back, so we know that 2 hydrogen ions, it forms 1 ATP. So for the 6 hydrogen ions, we'll get almost up to 3 ATP molecules. So hence for one NADH molecule, 3 ATP molecules will be formed. Whereas for the FADH2 molecule, the process starts from the complex 3, right? So electron will be accepted by the coenzyme Q. So hence here, the hydrogen released in the complex 1 won't be considered here. So here, if we consider for FADH2, the hydrogen ions will be released in the region of complex 3 and complex 4. So totally for one FADH2 molecule, 4 hydrogen ions will be released. As a result, it is coupled with the production of 2 ATP molecule. So that is regarding the amount of ATP that is produced upon oxidation of one NADH and one FADH2 molecule. So one NADH gives rise to 3 ATP and one FADH2 gives rise to 2 ATP molecule. By considering it, so next we'll calculate the respiratory balance sheet. So it gives an account of, so this is for the aerobic respiration. So we are going to calculate the respiratory balance sheet. So which gives an account of number of ATP that is produced. So in case of glycolysis, directly 2 ATP molecules are produced. So that is the net gain of ATP in glycolysis. In addition, so we have seen in the end product of glycolysis, 2 NADH molecule will be produced. So one NADH molecule, it produces 3 ATP. And for the 2 NADH molecule, so it will be 3 into 2, that is totally 6 ATP molecules will be formed. So hence, the total number of ATP produced in the glycolysis will be 8 molecules. And next is in the preparatory step or in the pyruvate oxidation, that is before the pyruvic acid is transported to mitochondrial matrix. So it undergoes oxidation to form a coenzyme A. And during this step again, 2 NADH molecules will be formed. So which gives rise to 6 molecules of ATP. And in case of the Krebs cycle or citric acid cycle, we have listed the end products. So 2 molecules of ATPs are produced directly. And totally in the reaction, 6 NADH molecules are produced. So for 1 NADH, it is 3 ATP. And for the 6 molecules of NADH, it will be up to 18 ATP molecules. And 2 FADH2 molecules are produced during the Krebs cycle. So which produces totally 4 ATP molecule. So hence here, if we look in case of Krebs cycle, the total number of ATPs produced will be, so it will be almost up to 24 molecules of ATP and 6 ATP from the pyruvate oxidation. And totally, if we consider the number of ATP produced at the end of aerobic respiration, it will be 8 plus 6, that is 14 plus 24. Totally, it will be 38 molecules of ATP. So that is how we are going to get 38 molecules of ATP at the end of the aerobic respiration. So that is regarding the respiratory balance sheet. So this is the total number of ATP produced. But if we consider the net ATP production, so it will be 36 molecules. They will consider only 36 molecules of ATP as a net gain in aerobic respiration. That is because the 2 molecules of ATP that are produced directly in the glycolysis. So this will be used in transfer of pyruvic acid into the mitochondrial matrix during the preparatory step. So these 2 molecules are considered as insignificant here because they will be utilized during the internal reactions itself. So if we deduct these 2 molecules, so the net gain of ATP will be 36 molecules of ATP. So in case of examination, NEET examination, the question with respect to the number of ATP in aerobic respiration can be asked in 2 ways. One is the total number of ATP produced in the aerobic respiration. So that will be 38, whereas net gain of ATP in the aerobic respiration, if it is asked, then it will be 36 molecules. So that is regarding the electron transport mechanism and the respiratory balance sheet. So in the next session, we'll discuss regarding the amphibolic pathway of the respiration as well as the respiratory quotient.